Welcome to the PowerPoint on blood. So let's talk about the contents of blood. First of all, if you centrifuge blood in a tube, you're going to see about 45% of what you centrifuge being red blood cells and what's called a buffy coat, which is composed of white blood cells and platelets. The other 55% is going to be plasma. Now, if we take a closer look at the components of blood, we're going to see a number of different things. First of all, when we're talking about the body weight of fluids, overall body weight of fluids, other fluids and tissues are about 92% of the body weight, and about 8% of body weight is whole blood. Now, in terms of the volume of whole blood, what's in here? Well, about 55% of it, as I said, is blood plasma. And what are the components of blood plasma? Well, by weight, it's about 91.5% water, about 7% proteins. And those proteins consist of albumins, globulins, fibrinogen, and other trace proteins. Other solutes include electrolytes, nutrients, gases, regulatory substances such as hormones, and waste products. Now, as far as the other 45% by volume, it consists of formed elements, which are cells and fragments of cells. Of those formed elements, in terms of the numbers per microliter, there are about 4.8 to 5.4 million red blood cells per microliter. And I want to remind you that a microliter is 1/1000th of a milliliter. So we're talking about a really small volume here, yet it still contains 4.8 to 5.4 million red blood cells. There are about 150 to 400,000 platelets per microliter and about 5,000 to 10,000 white blood cells per microliter. When you start to go above about 10,000, that may be an indication that you've got some sort of infection going on. And so when white blood cell counts are done, anytime that you see a count above about 10,000, alarm bells are going to start going off. Okay, so the different white blood cells are neutrophils, which make up about 60 to 70% of all white blood cells, lymphocytes, which make up about 20 to 25%, monocytes, which make up 3 to 8%, eosinophils, which make up 2 to 4%, and basophils, which make up 0.5 to 1% of the white blood cells in a given microliter. So taking a look at some of these components of the blood in this picture, here are examples of red blood cells. So you can see that by far they're the most common formed element. Here are white blood cells or leukocytes, and in fact, all of the leukocytes that you're seeing here on this page are neutrophils. The one on the bottom left might be a monocyte because the nucleus is colored a little bit differently, but it could also be a neutrophil. Here are examples of platelets. So platelets are fragments of larger cells called thrombocytes. And of course, the remaining space that you see on the slide is blood plasma. So we're going to talk about erythrocytes for a couple of slides, and erythrocytes are the majority of the formed elements, between 99 to 99.5% of what you see in whole blood. So a hematocrit is a measure of the percentage of red blood cells in whole blood, not a percentage of how many formed elements you have, but the percentage of the volume of red blood cells in whole blood. So 45% on average, as you saw in the tube in an earlier slide, is about the average for people. For males, it's about 42 to 56%, so 45% would actually be a little bit on the low side for males. And the reason why males have a higher hematocrit is because of testosterone, and testosterone will stimulate erythropoietin, which is the hormone that directs the formation of red blood cells. For females, a 45% hematocrit would be pretty high. And their hematocrit is lower because of female loss during menstruation. So let's talk about the shape and the size of red blood cells. First of all, the shape is a biconcave disc. And if you look at the bottom diagram, you'll see the sectioned view. And so basically, it almost looks like it's scooped out at the top and the bottom. The size of a red blood cell is about 8 micrometers. It has a central pallor, so if you recall looking at any of these red blood cells under the microscope, you may remember that the center of the red blood cell was a little bit lighter than the rest of it, and it's because it's thinner and light comes through it more easily. Red blood cells don't have a nucleus or organelles. They eject the nucleus and the organelles as a normal part of their formation. They don't use any of the oxygen that they transport because they're not involved in any sort of metabolism. So without the need without a metabolism, they don't really have a need for any oxygen. So they don't use any of the oxygen that they actually transport. Now this biconcave disc gives a red blood cell increased surface area, both for oxygen and for a carbon dioxide exchange. But I want to make it clear that red blood cells only carry about 13% of the CO2 that's carried in the bloodstream. So that's really much more of a minor function than carrying oxygen. So this biconcave disc shape gives them a huge surface area relative to volume. And so that allows oxygen to diffuse into the red blood cell much more easily than it would otherwise. They're basically bags of hemoglobin. Red blood cell is 97% hemoglobin. So each red blood cell will contain 250 million hemoglobin molecules. And given that each hemoglobin molecule carries 4 oxygen molecules, how many oxygens does that make in each red blood cell?
1 billion!
Okay, so now we're going to talk about leukocytes. So leukocytes actually make up less than 1% of the blood volume, and in a centrifuge tube, they're actually part of the buffy coat. Now, unlike red blood cells and platelets, these are complete cells with both nuclei and organelles. And if you want to remember the most common versus the most rare concentrations of white blood cells in the bloodstream, just remember this. Never let monkeys eat bananas. So that's neutrophils, lymphocytes, monocytes, eosinophils, basophils. Okay, so taking a look at white blood cells, we're going to look at what are called the granulocytes first. And so granulocytes have what are called cytoplasmic granules in them that show up with staining, and they are all phagocytic, at least to a certain extent. So neutrophils are your first responders within 90 minutes of an injury. Now, they're going to also die rapidly between 6 hours to a few days. Under the microscopes that we've got, you wouldn't be able to see their granules because they're clear and they're difficult to see. But the main characteristic of leukocytes that you can sort of pinpoint is this multi-lobed nucleus. So basically it looks like it's got 3 to 6 nuclei with just really thin strands of connecting nuclear material. So that's what neutrophils look like. Again, first responders, die rapidly, phagocytic. Eosinophils are parasite fighters, and they have red granules. And if you were to see one of these under a slide, then the granules actually look about the same color as the red blood cells that surround them. So their main function is fighting worms. So they're parasite fighters, and they're going to release proteins that are toxic to worms. They're going to swarm them when they get into the bloodstream. Now, eosinophils live about 8 to 12 days, and they're also really key in an inflammatory response, especially allergies. And their nucleus has got 2 lobes with 1 thin strand, and if you hold your hands up, kind of like you're making goalposts with your thumbs attached, that's kind of what they look like in terms of their nucleus. The final type of granulocyte that we're going to look at is basophils, and these get called allergy cells. So they've got this really dark purple, blue granules, and sometimes these can even hide the nucleus. Now, these granules contain histamine and heparin, and these increase blood flow during inflammatory and allergic reactions. And in fact, basophils are the white blood cell that are most associated with allergies. So at one of my stops, there was a slide on the blood smear, and it was pretty clear that whoever had given the blood for that blood smear was in the midst of an allergic reaction, because there were way more eosinophils and basophils than what you would expect. Okay, the agranulocytes don't have cytoplasmic granules, and so they're going to produce antibodies, and they're going to carry out phagocytosis. And so the first type I want to talk about is lymphocytes, and these can be killer or antibody cells. We've covered those sorts of topics with the immune system in lecture, so you know I'm referring to cytotoxic T cells and B cells. Now, these are the smallest white blood cell. They're about the same size as red blood cells. Now, under the microscope, a lot of the time people can confuse lymphocytes and basophils are much larger than a red blood cell. So that's the main difference that you can tell. So they've got a large nucleus that takes up most of the cell with a really thin rim of cytoplasm around it. So despite the fact that they're the second most common type of white blood cell in the bloodstream, only a small proportion of them are actually in the bloodstream because most of them are in lymphoid organs. In your lecture videos, we've discussed how some B and T cells can be kept back for memory cells, and this means that these can live for years. So the cytotoxic T cells are involved in direct cell killing of cancer cells and cells infected by virus, and the B cells will produce antibodies after maturing into plasma cells. The other type of a granulocyte is monocytes, and these are by far the largest white blood cell. Now they've got sort of this U or shaped or kidney shaped nucleus, and sometimes this can stain a little bit more reddish than the other nuclei. They've got a lot of cytoplasm surrounding them, and what monocytes do is to take over for neutrophils when they arrive at the site of an injury or an infection. And they're better at neutrophils than at phagocytosis because they're bigger, so they can take in larger objects and they take in larger quantities than neutrophils can. So although the neutrophils get there first and start to address maybe infection or debris from an injury, the monocytes are going to swoop in and take over. Okay, so thrombocytes or platelets provide a plug at an injury initially to prevent blood loss. And so they'll be prompted to do this when an injury to the vessel, to a blood vessel, disrupts the endothelium and exposes collagen. And so platelets adhere to collagen when they detect it, and then they'll release products from their secretory vesicles that activate other platelets. Now, when those other platelets settle down, they'll do the same thing. They'll release their products, attracting other platelets, and you can have a positive feedback reaction that could potentially get out of control. However, the cells that are next to the injury will produce chemicals called prostacyclins, and these will inhibit platelets. And this is how you try to keep a platelet plug localized instead of spreading beyond the bounds of the injury. Okay, so with blood plasma, we've already seen the components of this, but I wanted to tell you the function of the different proteins. So, and of water. So water absorbs heat really well, and that means that water doesn't change temperature fast in response to an outside temperature change. So with this stability in temperature that water can maintain, blood gets involved in thermoregulation. So other proteins, albumins maintain osmotic pressure. And basically what this means is that albumins serve as the particles that can pull water across membranes, more specifically across capillary membranes towards them. So as long as you have a certain concentration of albumins in the bloodstream, you should also have a certain concentration of water in the bloodstream. Globulins are involved in the immune response, and there are several different types of these that are part of the immune response, which we cover in the immune system videos. Fibrinogen is the precursor of fibrin, which is your main clotting protein, and we talked about the clotting cascade earlier this semester in lecture. And you get serum when you remove fibrinogen and clotting factors from blood plasma. Okay, so we're going to take a look at blood type next. So you've got two main systems for blood typing, ABO and RH. And let's take a look at ABO first. And so basically A and B are red blood cell surface antigens, and these get called agglutinogens because of the agglutination reaction, which we'll see in a few slides, that happens when you have mismatched blood donation. So if you're blood type A, then your red blood cells have A surface antigens. If you're blood type B, your red blood cells have B surface antigens. If you're type AB, you have both surface antigens, and this is the least common type, And if you're type O, which is the most common type, then you have no surface antigens at all. So you also have preformed antibodies in the plasma to either A or B, depending on what blood type that you have. So these are the antibodies that really are responsible for sticking red blood cells together. So if you have type A blood, you are not going to produce an antibody against the A antigen because you're not going to produce an antibody against what you possess. So if you're type A, you'll produce anti-B antibodies. So you produce antibodies against what you don't have. So if you're type B blood, you're going to produce type A antibodies. If you're type AB blood, well, you've got both antigens. You don't want to produce antibodies against either one of them, so you actually don't have antibodies produced. And if you're type O blood, you don't have either the A or the B antigen, so you're going to produce both anti-A and anti-B antibodies. And so the antibodies that you produce have real implications in terms of what type of blood that you can accept. So why is it that the antibodies are preformed with A and B? Why are they there? Well, they're present in our plasma prior to first contact with blood of a different type. And the reason why they're there prior to that is because the exposure that you get is that A and B antigens are very similar to antigens that are produced by bacteria and some plants. So we come into contact with the antigens produced by bacteria and plants that very, very closely resemble A and B, and so we can develop long-term active immunity to those antigens. And so because these same antigens are present on the surface of red blood cells of different types, then you've got antibodies present that will bind to red blood cells that carry foreign antigens, and this usually happens within 6 months following birth. So this gets really relevant when you require a blood transfusion because you don't want to get incorrectly matched blood that causes an agglutination. And so what happens to cause agglutination? You get the binding of antibodies to the red blood cell antigens, and then they stick together. So this agglutination or clumping results in the obstruction of blood vessels, for one thing, and the hemolysis of the red blood cells, of the donated red blood cells. And this is also a bad thing because, you might as well not be getting blood if you're bursting the red blood cells. Plus, hemoglobin, free hemoglobin in the bloodstream is toxic. So if you give somebody the wrong blood type, you get blockages of blood vessels, you don't increase their oxygen carrying capacity, and you're poisoning them. And so this is why it's so important to type and cross-match patients for transfusions. So continuing with our discussion about cell surface antigens, what's present in the blood plasma, what blood type that makes you, let's take a look at some of the different ABO blood types and see, the big picture of what's going on. So if you have antigen A present on your red blood cells, you are not going to produce an antibody against your own self-antigen. So basically, if you have antigen A on your red blood cells, you will produce anti-B antibodies because you produce antibodies against what you don't have. So if you're type A, you don't have type B antibodies. So you don't have type B antigens, so you're going to produce antibodies against them. This means that you're going to be type A. So erythrocytes with type A surface antigens, plasma containing anti-B antibodies. Now, if you have antigen B on the surface of your red blood cells, since you have antigen B and you don't have antigen A, you're going to produce anti-A antibodies. One of the reasons why I like this diagram is because it shows the shape of the antibodies and the shapes of the antigens matching. So if you look on the left at the anti-B antibody, you can see that it matches up shape-wise with antigen B. And if you look at the anti-A antibodies, you can see that they match up with the antigen A. So anti-A will match up with A, and it'll clump and agglutinate. So if you're type B, your erythrocytes are going to have type B surface antigens. You're going to produce anti-A antibodies in your bloodstream. So what happens if you have both of these antigens? If you have antigens A and B, what antibodies are you going to produce? The answer to that is none, because you're not going to produce antigens against what you produce. So that means that if you have antigens A and B, you don't produce any antibodies, and so your erythrocytes are going to have both type A and type B, and the plasma will not have anti-A or anti-B antibodies. Okay, so, well, that kind of gave the preview away. Um, so if you have neither antigen A nor B, this means that A and B are going to be foreign as far as your body is concerned. Therefore, you're going to produce both anti-A and anti-B antibodies. So again, the big thing that I want you to remember here is anti-B binds to B, anti-A binds to A. So this is going to mean that your blood type O, so you don't have type A or type B surface antigens, but your plasma will have the antibodies against both A and B. So you're going to have anti-A and anti-B. So what's the point here in terms of blood transfusions? Well, if you're type O, your cells are not going to cause a reaction if you put them into somebody else's body. So since your cells, if you're type O, don't carry antigen A or B, this is going to mean that you can donate to type A, type B, or type AB. Now, if you're type O positive, this means you're going to be able to donate to about 85% of the population. If you're type O negative, you basically got golden blood as far as the blood donation services are concerned. You can donate to virtually 100% of the population, unless you've got a conflict with some of the rarer blood groups that we'll see a little bit later on. So again, if you're type O, you're a giver. You can give to anybody. Can you receive blood from other blood types? The answer to that is no. Because if you're type O, if somebody puts type A blood into you, you've got anti-A antibodies, you're going to get agglutination. If you put type B blood into you, if you're type O, you're going to have antibodies against B. You're carrying around anti-B. So that's going to agglutinate. And type AB is going to be probably the worst thing you could get, because you have both anti-A and anti-B antibodies. Both of those are going to react against type AB blood. So you can give to just about anyone, but you can't receive from anyone but other type O people. Fortunately for our blood supply, type O is the most common blood type. So that's great, because it means that type O people will struggle less than they might otherwise to get compatible blood, but it also means that there are more people out there who can donate to a variety of individuals. Now, if you're type AB, you can only donate to other people who are AB, but you can accept anything. So if you're type AB, you're kind of a taker. You're not a giver, you're a taker, because you can take any type of AB antigen blood. So again, if you're type AB, it's good to, I suppose it's good to go in and donate, but you're going to be a much better recipient than you are a donor. Okay, so as far as agglutination itself goes, let's take a look at this. And so this is the mechanics of how it happens. So you're going to see an antibody binding to the antigen of a foreign cell. Okay, so if you take a look at that foreign cell, look at the little dots that are sort of present on the surface of the cells. So you see the cell surface protein being one shape in the three cells to the top left, and the foreign cell to the bottom right has a different shaped antigen on its surface. And that antigen, unfortunately, matches the antibody that the self person has produced. So what happens is these antigen and antibody complexes start forming, where red blood cells actually stick to antibodies and they get stuck to one another. And this is where agglutination or clumping actually happens. So if we take a look at how you test for this. Now, the very first thing that I want to tell you is that what you're going to do here is you're going to put blood samples down into wells or tubes that contain the antibodies. So you can see that anti-A and anti-B are already present here. Okay, and you're adding blood to that. Now, if you add type AB blood, you're going to have both A and B antigens, and those will react against both anti-A and anti-B, and you can see that clumping there. If you're type B, then you contain the antigen or agglutinogen B, and if you introduce that to pre-existing antibodies, you'll get no clumping with anti-A because you don't have antigen A. You will get clumping with anti-B because you have antigen B. So again, I want to remind you that type B has antigen B, which will react against anti-B. If you're type A, then you contain agglutinogen or antigen A. And so what's going to happen with the blood serum here, you'll react against anti-A and you will have no reaction against anti-B. If you're type O blood, you don't have either A or B, so when you introduce type O blood to anti-A and to anti-B, you get no reaction with either one. So the agglutination simply doesn't happen at all with type O because it doesn't contain the antigens that cause agglutination against anti-A or against anti-B. So let's say that you have a donor that is type A, and you have a recipient who is type A. They contain anti-B antibodies, but that's no problem because you don't have type B blood that's being donated. So type A donating to type A means you get no agglutination and no clumping. So this is a successful blood type match. Now, if you take blood type A and you donate it to blood type B, if you look at the shape of the antibody of somebody with blood type B, they have antibodies that will clump with antigen A. And so what you will see here is agglutination because the antigen and the antibody, they match and they'll bind to one another. And this is when you can see clumping happen. This is when you see hemolysis occurring and you have an unsuccessful blood type match. So all of this is basically a transfusion reaction when incorrect red blood cells from the donor get destroyed by the antibodies of the recipient. And so again, a type B person has anti-A antibodies. So if you give them type A red blood cells, you're going to get agglutination. So we use this agglutination in order to do blood typing because we definitely would rather have this reaction occurring on a slide or on a well outside the body rather than inside the body. So this is what it could look like if you get the wrong type of blood introduced. So if you look at the redder red blood cells, then you'll see that this is the type B blood. These are the type B blood cells. And the erythrocytes for type A are colored purple just to kind of distinguish them. But you can see at the bottom text where you've got the agglutinated erythrocytes blocking small vessels. So you can get blockages. And again, what this doesn't show also is the hemolysis where the purple red blood cells would be bursting. Now, I don't want to give you the idea that type A and type B blood types have different color blood. That's just something that's done for emphasis in the slide. Okay, so the other type of antigen system that we look at is the Rh antigen system. So we've been looking at A and B and O. But if somebody asks you what type of blood that you have, you wouldn't just limit it to, I have type A blood. You'd want to say type A positive or type A negative. And the positive and negative are actually an indication of whether or not you have the Rh antigen. So again, Rh is another red blood cell surface antigen, and it sometimes gets called antigen D. So there are actually 8 types of Rh antigens, and common types are C, D, and E, but it's either present or it's not. So if it's present, you're type Rh positive, and this is 85% of people, and if it's not present, you're Rh negative. So if your erythrocytes have antigen D, then you don't have any anti-D antibodies, and your blood type is going to be Rh positive. So if you don't have the Rh antigen, then you don't have any antigen D, and you will form anti-D antibodies, but you will only form these upon first exposure, because if you'll recall, the A and B antigens have got very similar antigens that occur out in the environment. Antigen D does not have similarly occurring antibodies in the environment. So this means that the only way that you actually form anti-D antibodies is if you have the wrong type of blood introduced. So your erythrocytes are not going to have any type D surface antigens, and the blood plasma will only have antibodies if there's been prior exposure to Rh positive blood. Now, what does this mean in terms of blood donation? It means that if you're Rh negative, then you can donate to any Rh positive person. It means if you're Rh positive, you cannot donate to an Rh negative person if they've had prior exposure. Fortunately for us, in terms of blood donation, Rh positive is about 85% of the population. So my blood type is O positive, and I can donate to any ABO blood group. And because 85% of people are Rh positive, it means that I can donate to about 85% of the population, which means anytime that I donate, either the Red Cross or Medic, or they're just all over me after I donate, asking me when I can come in and donate again. So again, no naturally occurring antibodies to the Rh antigen, because again, they only develop if you're exposed to Rh positive blood. Okay, this only happens either in an incorrect transfusion, or it can happen in what's called Rh incompatibility of pregnancy. So this is what happens if an Rh mother has a child with a father that's Rh positive, and if that first child comes out Rh positive, and that's pretty likely because positive is dominant to negative, then the child is fine, but this first exposure may cause the mother to develop anti-D antibodies, so Rh antibodies, upon exposure to the infant's blood. Now that doesn't cause a big problem with the first pregnancy, but it can cause big problems with subsequent pregnancies. So if you look at this first pregnancy, it's pretty common for fetal blood to get out into the mother's circulation. And so that means that these Rh positive antigens can get out into the Rh negative mother's bloodstream. And so if you look at the bottom left picture, she's going to form anti-Rh agglutinins. This becomes a real problem if you have a second Rh positive fetus, because those anti-Rh antibodies can cross the placental barrier and cause serious damage to that second fetus. This is actually one of the reasons why it used to be legally required for you to have a blood test before marriage, because of this. Okay, so again, with a second pregnancy, the mother's antibodies can cross the placental barrier and bind to the Rh antigen in the red blood cells of the fetus, and this leads to bursting of the red blood cells in the fetus and what's called hydrops fetalis. Now, how do you prevent this? It's usually a good idea, if you don't already know the blood type of the person you're having children with. If you're an Rh negative woman, then you need to find out, if you're planning on having children, the blood type of the man you're having children with, because you can get a shot of what's called RhoGAM, and what RhoGAM is it is a preformed antibody to the Rh antigen. And so you may be thinking, if these antibodies can potentially cause damage to the fetus, why have these injections? Well, what you're going to do is you're going to inject them at a low enough concentration to not damage the fetus, but at a high enough concentration to encounter and burst the fetal red blood cells that make it into the maternal bloodstream. Now, one of the things that you learn in the immune system chapter is that if you are given antibodies, then you don't form your own antibodies and you don't retain a memory of that particular antigen. So basically what this does, these RhoGAM shots, is they protect a second pregnancy from encountering these antibodies. So basically, if a mother hasn't been sensitized, she gets one injection at about 28 weeks of pregnancy, and then within 72 hours of either a birth, a miscarriage, an abortion, or an amniocentesis. And so again, these antibodies destroy any red blood cells from the baby that have entered the mother's blood, and so she doesn't get sensitized to the Rh antigen, and she doesn't form memory cells against the Rh antigen, and the Rh incompatibility shouldn't be a problem during the next pregnancy. But this does have to happen with each pregnancy, the RhoGAM shots, if an Rh negative woman has children with an Rh positive man. Now, if you're female and you're Rh positive, you don't have any problems, because if you're Rh positive, then the fetus is going to be Rh positive, and it doesn't matter if your partner has Rh positive or Rh negative blood. You're not going to produce antibodies against the Rh antigen, and so it doesn't really matter if the fetus is actually Rh positive or Rh negative. So there are other blood types besides ABO and Rh, and there's a lot of them. So these aren't as commonly assessed because they're not as common as the ABO and Rh blood types. And these aren't even all of the other blood types. There are 34 other blood group systems with more than 300 known variants. So of these other blood types, even though there's a lot of different blood group systems in total, ABO antibodies are clinically significant for transfusions, but other groups that can cause transfusion reactions besides ABO and Rh are going to be the MNP groups, the KEL and KX blood group systems, the KID blood group system, and the Duffy blood group system. So these can be clinically significant, and and you can be typed for those blood systems as well as ABO and Rh.